So today we'll discuss the kingdom fungi. This is the third kingdom. We have already discussed kingdom Monera and kingdom Protista. So now this fungi it constitutes a separate kingdom in Whitaker's five kingdom classification. So which type of organisms are included under it? These organisms are eukaryotic. Remember, eukaryotic and heterotrophic. Okay, and they show greater diversity in shape, size, and habitat. They show greater diversity in shape, size, and habitat. So they dwelt in varied habitat. They have various types of shape, size. Okay, and the common examples as per your NCERT, the common examples are mushroom, that you all know about it, then bread mold, yeast, rosts, and smots. Okay, so these are the some of the common examples that you know about. Uh, Now, what are their diversified habitat? As I said you, so they are found in air, water, soil, and also on plants, soil, on plants and animals. And they preferably they grow in warm and humid places. Okay, so for their growth, they always see, if you keep a piece of bread in the deep freeze, then you will see that the fungus are not growing. But if you will keep it in the or outside the refrigerator, then very soon you will see that the fungus are growing on it because of the warm condition. Okay, and but inside the refrigerator it is getting the moisture, but it is very cold, that's why it will not grow, the fungus will not grow. Okay, now let us see the cell structure of the fungi. They are eukaryotic. You know the features of eukaryotic cell, so I'm not discussing that one. Now, uh, previously the Monera were having they were all prokaryotic, but the Protista were eukaryotic. So, eukaryotic means they have a true nucleus and membrane bound organelles. Now, the most important is their cell wall is made of, very important, chitin, very very important, which is a polysaccharide. You know what is polysaccharide? Polysaccharide is a type of carbohydrate. I hope you have studied it in your previous classes. If you don't know about it, you will discuss. We'll discuss in the chapter biomolecules. Okay. So for the time being, you just remember that polysaccharides are the carbohydrates. Okay. Now coming to the body organization. So most of the fungi, almost all fungi are multicellular. Mostly multicellular. But exception is there. Exception, that is yeast, is unicellular. Very, very important, children. Fungi are multicellular, but yeast is unicellular. Now, regarding their body organization, in more details, the body consists of thread-like structures called hyphae. Okay, very very important. I'm underlining these points. Now this network of, I will show you the diagram. Network of hyphae forms the mycelium. Okay. Now let me show you what is mycelium and what is hypha. Now see, okay. So the whole structure is mycelium. Okay, the whole structure, uh, the network of a hypha. These are the hypha. So these hypha are the thread-like structures which together constitutes the mycelium. Okay, so this whole structure is the mycelium which consists of the hypha. Okay, so now these hypha are of two types. Now come to the hypha. One is cynocytic, or you can say they are aseptate. Okay, and other one is septate hypha. Okay, so what are these two structures? What is the difference between these two? See, I have shown you the hypha. Okay, so here also, okay, now I'm coming to the diagram. Now let me tell you the difference. They don't have the sinocytic or accepted hypa. There is no septa. Septa means what? Cross wall. Okay, and the cytoplasm is multinucleate. Multinucleated cytoplasm. And because it is multinucleated, so it forms a continuous tube like structure. Okay, now come to the diagram. See, as you know, it, as I have said you, it is a multicellular organism. So it consists of many cells. But here what happens? The cells are not separated by the cell wall. There is no cell wall. So because of which here you can see that you will see that the nucleus, or you can say the cells, are not separated by the septa. Septa means this cross wall. So this cross wall is absent. Okay, so that's why the cytoplasm is multinucleated. The cytoplasm of each and every cell is connected. It is a continuous cytoplasm, and the nuclei of different cells are present in a single cytoplasm. So it is known as multinucleated cytoplasm. I think you it is clear to you. Once again, I am repeating. The hypha, as I have said you that almost all fungi are multicellular. So this is also multicellular. So cell means what? Cytoplasm is present. In the cytoplasm, the nucleus is present and the cell is surrounded by the cell membrane. Okay. So this should be the structure of a cell. Now here what happens? This cross wall is absent. Since this cross wall is absent, so the peripheral cell is here. So there is no wall between this. Then what happens? The cytoplasms are continuous, and in a single, it forms a single cytoplasm where more than one nuclei are present. So I think it is clear to you what is accepted. Accepted means no septa wall. Septa wall means cross wall is absent. Now come to this septate hypha. So in septate hypha, cross walls or septa present. Okay, and because the cross walls are present, so the cytoplasm divided into compartments. Okay, so now come to this diagram. So here you can see that the cytoplasm of the cells of hypha are separated by the cross wall. Okay, so here the cytoplasm is not continuous. A septa or cross wall is present, so it is known as uh, um, your septate hypha. And here, note down, children, the very important point: the sinocytic hypha are characteristics of lower fungi. That means the simplest fungi, that is Phycomycetes. So we are coming to this Phycomycetes. So this is very important point, children. So this is the characteristics of Phycomycetes. Now come to the more, we are discussing the general feature here, not the specific feature. So now the mode of nutrition. So they don't have chlorophyll, so no photosynthesis. Okay, so there the various types of nutritions are they are mainly saprophytic, and you know what are saprophytes? They feed on dead and decaying organic matter. Okay, and they are the important consumers. Okay, because they digest the food, convert the food into soluble form, then they absorb the soluble food from the organic matter. So for example, bread mold, mushroom. So these are the common examples that you know. Okay, now there is another type of nutrition, or you can say the relation they establish with other organisms, that is parasitic. This is saprophytic. The next one is parasitic. Means they obtain food from living organisms, other living organisms, and such type of fungi cause diseases in plants and animals. Okay, for example, rosts and smots. So they cause disease. These two fungi, parasitic fungi, they cause disease in plants. Okay, now the third one is symbiotic. They establish symbiotic relationship to obtain their nutrition. So in symbiotic relationship, they live in mutually. All of you know what is symbiosis, because you are well known about the lichens. Mutually beneficial association with other organism. Okay, so here the again here we'll discuss about the two symbiotic relationship of fungus. That is, first one is lichen. Okay, so this is the association between fungus and algae or cyanobacteria, means blue green algae. So here in this relationship what happens, algae prepares food, and because it is mutual relationship, so from the algae the fungus gets the food, and the fungus provides, in turn fungus provides shelter and water to the algae. Okay, so here the in this symbiotic relationship, in case of lichen, they are associated with the fungus, are associated with the algae or the cyanobacteria, where the algae are autotrophic for photosynthetic, so they can prepare their food, and this food is provided to the fungi. But because it is mutual relationship, in turn fungi provides shelter to the algae. Okay, now the next relationship, that is symbiotic relationship of fungi, is mycorrhiza. So this is the association between fungi and root of higher plants. Okay, so this is also a mutual relationship. So in this relationship, how the fungus help the trees or the plants, the fungus helps in absorption of water and mineral, and in turn plant provides food. So these are the different types of mode of nutrition. So in mycorrhiza what happens, this is a big tree and here is the root. So the roots are not so branched, but when it is associated with the fungus, you know that the fungus are having hypha, so the root becomes more branched due to the association of the fungi. So as it is more branched, so it absorbs more water and minerals from the soil. Okay, so this is the way how the fungus helps the plant. Now come to the reproduction in fungi. Okay, now this fungi can reproduce by three methods. First one is vegetative reproduction. So the vegetative reproduction takes place by three methods, that is fragmentation, then number two fission, and budding. Okay, so for example, yeast reproduce by budding. Okay, so fragmentation, you know, that is accidentally, that is when the fungi is fragmented or it is divided into pieces, then each piece develops into a new fungus. Now fission, you are well aware about the binary fission, multiple fission. So this is also that type of fission, means division of the hypha. Okay. Then budding, you are well known about this budding, and this takes place in case of yeast. Now come to the next type of reproduction is asexual reproduction. Okay, so this asexual reproduction takes place through spores. Okay, and these spores are of different types. Different types of spores are there. So now let me tell you types of spores. And the detail about the spore is not given in your textbook, but let me tell you because it comes in your exam. Okay, uh, the spores are of three types, that is conidia or conidiospore, then sporangiospore, and the other one is zoospore. Okay, so what are the differences among these three? Now conidia are, let me first write down the features, then I'll show you with the help of explain you with the help of diagram. So the conidia are non-motile. Okay, so these are asexual spores, so I am not repeating the term asexual spore. Okay, now sporangiospores are, I'm sorry, produced inside specialized sac called sporangium. Okay, and they are non-motile. Okay, but the zoospore are motile. Now next point, they are formed, conidia are formed externally. Okay, usually at the tip of specialized stalk called conidiophore. Okay, very very important. And sporangiospores are developed internally inside sporangium. Okay, and the zoospores are also formed internally inside zoosporangium. Okay, and they are released to swim in water. Now how do they, if they are motile, then how do they move? So I have already told you that these are non-motile, and so they cannot move. They are also non-motile, but the spores are carried by the wind from one place to another place. So these are actively motile. So why they are motile? Because they are having flagella. Okay. Now come to the diagram. Let me show you the diagram. The first one is conidiophore. So conidiophore, as I have said you, it is a stalk. The stalk is called conidiophore. The stalk is branched like this. The structure is like this. Okay. So these are the conidia. Okay. And this stalk is known as conidiophore. Okay. Now come to the sporangiophore. So it is a sac-like structure. Sporangium is a sac-like structure, and inside this the spores are present. So these are the spores, and these spores are non-motile, but they are released from the sporangium and they are carried by the wind. Now the last one, that is zoospore. As I have said you, they are motile. They are produced internally. Okay, so these are the zoospores, and they are having flagella, so they can move from one place to another place. Uh, Now let me tell you the examples. These conidia are seen in penicillium, and the zoospores are seen in chlamydomonas, and the spores are produced in bread mold. Okay, so these are the different types of asexual spore. So what are the general feature of the spores? The features of asexual spores. They are produced in large number, help in rapid multiplication, and dispersed by air or water. Okay. Okay. Now coming to the sexual reproduction. The third one is sexual reproduction. They also reproduce sexually by different types of spores. The sexual spores are oospores, ascospores, and basidiospores. Okay, so these are the three types of spores by which the different group of fungi reproduce. Now let me tell you the general method of sexual reproduction. The steps of sexual reproduction. We'll discuss about these spores while discussing the different groups of fungi. Okay, the first step, step 1, is plasmogamy. It is often asked as a 3-mark question, children. So in plasmogamy, what happens? The fusion of cytoplasm of two gametes takes place. Okay, so here what happens? Suppose the hypha, the each cell of the hypha may act as a gamete. So suppose say this is a gamete and this is a gamete. Their nucleus is here. So during plasmogamy, what will happen? During plasmogamy, the cytoplasm will fuse. Okay, so this is called plasmogamy. Now the next step 2 is karyogamy. So here what happens? Fusion of nuclei. Okay, so here you can see that the single cytoplasm is having two nuclei. Okay, so this phase is known as dikaryo phase. So what is this condition? This condition is the cell with two nuclei. Okay, this condition is called, that means because this is the formed by the fusion of the gametes, and gametes are always haploid. So this is n plus n, and this phase is called dikaryo phase. Very, very important. This phase is called dikaryo phase. And now what happens? After the fusion of the nucleus, the cell is having a diploid. So here the cell was n plus n. Now it is 2n. Okay. And after this, in the third step, step 3, meiosis. Okay. So now whatever structure is formed, now this is called zygospore. I'm sorry. This is zygote. After the fusion of the gamete, what is formed? It is zygote. Now this zygote undergoes meiosis and it forms haploid zygospores. Okay, and this is the characteristics of um, uh, almost all the fungi. But here the most important point you have to remember, in ascomycetes, and basidiomycetes, plasmogamy is not immediately followed by karyogamy. Okay, and this is all about the general characteristics of the fungi. In our next session, we'll discuss about the classification of fungi and other parts of this chapter. Okay, till then, bye bye. Thank you.